So here we are doing lesson number 11, neural signaling for SL, as part of subtopic C2.2. So we're going to be talking about neurons, how we make an action potential, a little bit about synapses, and different things that can affect the speed of neural signaling. So it's a long lesson today, so listen up and take notes, then we won't have to get behind. So first thing first, the nervous system of humans can be divided into two main parts. We have the central nervous system, and then we have the peripheral nervous system. So of course, the central system is just the brain and spinal cord. It's going to be the job of collecting all the information and then sending signals out to the rest of the body. And the peripheral, which here is kind of shown in yellow, are all these nerves all throughout the entire body. Now, what's the function of each of these different nervous systems? Take a minute and think about this. So the peripheral is going to be collecting information and responding to signals from the central system. Things like when your body is told you're too hot, time to sweat. It sends signals to your sweat glands. And also collects data about things like what temperature is the body at with thermoreceptors. So it's both those things. And then the central system is going to collect all that information and respond to it. The structure of each one helps support, and it's neat to be aware always in physiology of the connection between structure and function. So the structure of a system helps support its function by having, for example, a peripheral system has all these nerves all over the body, so we can detect signals all over the body to collect data on temperature, different kinds of set points for homeostasis, and then it can also respond to signals from the brain all over the body, tell all the parts of your body to do what they need to do to keep your body functioning correctly. We can also look at the nervous system as peripheral versus central and kind of break it down further. So central, of course, the brain and spinal cord. Peripheral here, all the nerves in yellow. Within the peripheral, we have two main sets of nerves. We have autonomic, which are things that are going to be self-regulated that we're not consciously controlling, like our pancreas secreting insulin to help us meet set point for blood glucose level. We're not telling it to do that. It's happening automatically. And then we have somatic, where we have voluntary control, things like skeletal muscle. You're choosing to move your body however you are, moving your hand, take notes. You're choosing to do that. And then we have the sympathetic and parasympathetic parts of the autonomic system. This is not required for IB, but if you would like to, find it interesting, you want to learn more about it, there it is for you, okay? So within the nervous system, the main cell we're talking about, of course, are neurons. Neurons are long cells, and they have evolved to transmit electrical impulses. The way that signal works within a neuron is electrical in nature. So most cells in living things are going to be the same kind of size. They don't get too big because, of course, of surface area to volume ratio. We want to have a large surface area, lots of membrane, lots of things come in and out of cells for a relatively small internal volume, what's going to require things coming in and out for metabolism. But neurons are different. They vary significantly between species. The biggest neuron in a giraffe is significantly longer than the biggest neuron in a mouse. Why? Why do we think this is? Well, this is because the length of the neuron depends on how long the signal has to go, and it all has to connect from the central nervous system to the peripheral. So on humans, one of our longest nerves is called the sciatica nerve. It's made of many neurons, and it goes from the base of your spine down your leg into your toes. So people actually have issues with that particular nerve. And then a giraffe, it has to go from its base of its tail, end of its spinal cord down to its leg. It's a much longer distance than in a mouse. Right, so if we're connecting peripheral to central via a bunch of neurons bundled into a nerve, and therefore they get longer with bigger organisms. There are different kinds of neurons. You can see 3 different ones in this picture here, but we're mostly going to be talking about this style here. Multipolar is the most common type, so we're focusing on that in this course. But all neurons have 3 main parts that are always present. Take a minute, look at the diagrams, think back to your reading. What 3 main parts are there? Well, we have the dendrites, which you can see here. They are parts where this neuron is going to collect signals from another cell. We have the cell body here, which has a nucleus. The pink thing is the nucleus, and it has all the organelles the cells need to be able to function, like mitochondria to get energy, all that kind of stuff. And we have the axon, which is the long branching part that can get very, very long in long nerves, and that's a part that sends a signal down. We'll focus on when we talk about action potential later on. Now, nerve impulses always move in one direction. What direction do you see in this picture? Well, they always start at the dendrite, the signal's being collected, go to the cell body, and then down the axon at the other end. These end down here are actually called the terminal buttons, or the synaptic cleft, which we'll get to later on today. But always the same direction of flow. Okay, here's your first key ideas. Take a minute, read them. If you have any questions, make a note so you can ask me later or ask a classmate. Okay, so you have been given a printable for the structure of neurons and we'll be using this diagram a lot for this first part is the neuron part, which looks like this. The second part is the synapse part, which we'll look at later on in this lesson. Please take a minute, glue it all together into one big foldable diagram, and you can fill in the boxes as you go. Okay, now not all these parts are super important for SL. I have put stars for the ones that really matter, but you should have a general idea of how everything works. Okay? So here we go. First part, we're going to start with number 11, the dendrites. Just copy this information either word for word if you want to, or you can paraphrase it. That's totally fine. You'll have access to the slides and this video if you need to go back later. So dendrites are the branch projections off the cell body, and they're really where the neuron is going to collect signals from other neurons, okay, from other neurons' axon, through what are called synaptic transmissions, the chemical transmission we'll talk about later on today. This is called the dendritic tree, and it gives a big surface area, all those branches, for lots of signals from other neurons to help connect. And one of the ways we can tell how much someone's using part of their brain is by how many connections there are between different kinds of neurons. The next one, axon hillock, that's more important for HLs, but SLs you can definitely talk about it as well. It's a part of the cell body that connects to the axon, and this is where the potentials from the dendrites are going to be what's called summated. It's basically where the axon says, okay, I got this many go signals, this many stop signals, what should my response be? Because your neuron's collecting signals from multiple other sources as well. Will it cause an action to happen or not? We'll talk more about that with HLs later on. Then we get into Schwann cells. These are important cells that are not part of the neuron cell. They're separate cells, but they wrap around the axon, okay? The myelinating type is a little bit important for everybody, but the name Schwann cells isn't that important for SL, okay? But the ones that are myelin, they're going to wrap around the axon, particularly of sensory neurons and motor neurons. So sensory neurons are those that connect to our sense organs, and motor neurons are those that connect to our movement organs, like our skeletal muscle. And they produce this myelin, which everyone needs to know myelin. And the myelin sheath is made of all those little myelin cells together, covering the entire axon. And it's really important for increasing the speed of nervous transmissions, which we'll talk about more later. So pause it if you need to, write down information, key ideas in these notes. The big ones for SL are dendrites. HLs, of course, need everything. And then Schwann cell, for SL, at least know that you have that's made of myelin, okay? Okay, the axon terminal, everyone needs that one. So these are the end of the axon. They're also called synaptic boutons, synaptic buttons. I like to call them synaptic buttons because they look little buttons to me. And they're between the end of the axon and the surface of the next neuron or the cell that the neurons connect to. If it's a muscle, if it's a motor neuron, that's to a muscle cell. If it's one for a gland, it connects to a gland, okay? It has a synapse that's created by making the gap between that neuron's axon terminal and the next neuron. Okay, it's called a synaptic cleft, a little gap between them. And the synapse is the neuron sending the signal, the small gap, and then the neuron receiving the signal. Again, we'll talk about this in detail when we talk about the synaptic part, which is the second part of your big diagram later on. Okay? And they do this transmission using neurotransmitters. The one we're going to talk about is called acetylcholine. Then we get to the cell body, which everyone needs to know. This is the part that contains nucleus and most of the organelles that the neuron needs to keep itself functioning properly. The nodes of Ranvier, these ones are more important for HL, but they're the gaps in the myelin because HLs have to know how that works. SLs just have to know that myelin makes things go faster, but HLs have to know why. So they're little tiny gaps between the Schwann cells. They're about 1 micrometer or micron wide. And that is where the impulse kind of jumps to. Again, we'll talk about that much more with HLs in the next class, and that's called saltatory conduction. It helps increase efficiency. So again, pause a bit if you're trying to write stuff down. These two are important for everybody. This one's just for HL. Okay, myelin sheath for everybody. So this is all the neurons that are myelinating, all the Schwann cells that are myelinating around the axon. And they're going to be important for helping make the signal happen more quickly. This is really important in our sensory and motor neurons especially. And then the axon itself is that long bit of cell projection that can vary significant length between species. And it takes electrical impulse away from the cell body and towards either another neuron or an effector like a muscle cell or a gland. They can be myelinated. We're going to focus on myelinated ones because they're the most useful ones, but they can also be unmyelinated. Depends on the cell. Okay, we'll talk about ones in giant squid later that are not myelinated. Okay, so here is your neuron structure, all the key parts. This is not the same diagram you have, but it goes through. So cell body that has the nucleus and all the important organelles. Then we have the myelin sheath. So SLs to know the myelin sheath, which is all of it. Only HLs have to live apart, so like the Schwann cells and the nodes of Ranvier, the gaps between. Okay, we have dendrites that collect the signals, and they have a large cerebellum branching to help increase the signal transmission. We have the axon, the signal travels down, getting to terminal branches or terminal boutons, or buttons if you want. Okay, all those things help with our key structures of the neuron. So make sure your diagram is clearly labeled. You can use some color to help you. It's a good idea, and make sure you have that good diagram for your notes. Okay, now I want to pick a minute and clarify the difference between a neuron and a nerve, because students often mix these two up. So neurons are individual cells. So here is the axon of a single neuron. Okay, but most nerves in our bodies are made of many neurons all connected together. Okay, so we can see in the picture here, we can see this is a peripheral nerve, and it has inside it blood vessels and then bundles of neurons. So all these bundles, every single segment is a separate neuron. Okay, our body contains between 95 and 100 billion neurons. Most of those, of course, are in our brain. Our brain is just a giant mesh of neurons. It's highly organized, but all our nerves in our body are also made of neurons. And the number of nerves actually is a little bit different between individuals. So what we call medically known nerves are ones that pretty much everybody always has. It's very rare someone doesn't have that nerve, but 214 of them. But our estimate for total number of nerves in individuals goes from 1,000 to 1,000,000. It's a huge variation because it's really hard to get actual data on that, and it's not that important, we don't think, to have the information. So for example, if you've been to the dentist and you've ever had freezing done in your teeth and your mouth, that's because you have nerves there. And I once had a filling where I had a surprise nerve. Then it's never seen anybody with a nerve in that spot before, and therefore she forgot to freeze it because didn't know it was there until I jumped out of the chair when she started drilling. And then realized, oh, you must have a nerve there, and then she froze it. But not everyone's nerves are in the same place, right? So it's a little variation, but nerves are made of neurons. Neurons are individual cells. Nerves are bundles of neurons. Okay, again, key ideas about neuron structure and function. Okay, let's talk about actually sending a signal along a neuron. So the first thing we need to talk about, and for us as we're going to focus on, is the resting potential. So when the neuron is at rest, the word we use for that is polarized. It's not sending a signal. It's resting potential, and we say that it's polarized, which means it has a negative charge inside the axon. In this picture here, we can see here's the axon, and it says negative charge inside and positive outside. It's the inside charge that matters for the signal. So when it's negative like this, at negative 70 millivolts specifically, it's at rest. There's no signal happening. Your body isn't telling it to do anything at that moment. So the reason why it's low, why its charge is negative, because we have a low number of potassium ions, or K+ ions, and a high number of sodium ions inside the fluid. Okay, so potassium ions are going to all be outside here mostly, and sodium ions are inside, but there's not that many of them. And then, but those are both positively charged, so how could it be negative? The reason why it's negative is because there are big proteins inside the axon that are negatively charged, and they are not able to move in and out of the cell. They're stuck. Only the small ions, potassium and sodium, can move in and out of the cell. So because the potassium ions inside the cell are too low in number to outcharge the big proteins, it leads to a negative charge. The proteins can't move, so when we change the charge, what we do is we change the ions location, put more ions inside that are positive until it's enough to outbalance the negative 70 millivolts of the negative proteins. The way I always remember this myself, and it might be helpful to you, is I think of the axon as a salty banana. We have sodium outside and potassium inside. Bananas are full of potassium, and salt is sodium and chlorine. So sodium and potassium, salty banana. That's the resting, okay? So it rests this way, and when a potential goes through, it will change. We're going to talk about how it maintains this resting potential by using the sodium-potassium pump, which we did talk about when we talked about membrane proteins earlier in the course. So here we go. So to reach this polarized state, this negative spin-volt charge, and be at rest, it has to use lots of ATP to pump sodiums out. So sodiums will rush in when it's sending signal, and to get it back to being at rest, we pump 3 sodiums out and then 2 potassiums in. Remember, salty banana. So sodiums out, potassiums in. And it's really efficient pump because it pumps out 3 sodiums and 2 potassiums for 1 ATP. So this is active transport because we're going against the concentration gradient. You can see the picture here, we're going against the gradient, but we're doing so pretty effectively by doing 5 ions for 1 ATP. Okay, so how a nerve impulse works here is we have the axon here, and you can see the resting or polarized area is pink and it's negatively charged. Now, when we're going to send a signal along the neuron, that's going to be changing that negative charge to a positive charge. So the positive green depolarized area, that's the charged area. So it starts here and then moves down and down and down. The entire axon is not depolarized or positive charged at once. It goes bit by bit by bit, okay? And then it goes back to polarizing after. This is really important. Think of it as a wave of positivity moving through the axon, because it's going to allow the signal to continue and then reset itself relatively quickly so another neuron can signal again. Like think about as you're moving your hand to take notes, you're not making one small movement and then stopping. You're continuously moving, so your neurons have to send these signals and then reset pretty quickly. Okay, let's talk about things that can affect the speed of a nerve impulse. So with that noise, guys. So different factors affect the speed of transmission. So the length of the axon and whether it is myelinated or not. There's 2 main factors we're talking about today. Okay. So here we have a myelinated axon and here we have an unmyelinated axon. They both work in the same way. This one is just way faster. So giant squid, here's the giant squid image here. They have a really large axon because remember axons go the length of the organism from essential to peripheral. So here's the brains go all the way down here in the front and all the way into the legs as well. It's up to 1 millimeter in diameter, which is really, really wide for a neuron, for a cell in general, and several centimeters long. So these projections of this one neuron allows the cephalopod, or the giant squid, to control its legs for propulsion, its water jet, all this kind of stuff. If you were here in 10th grade, you just said a squid, you've seen water jets and all those different structures. This is about 500 times bigger than the average human neuron. So its increasing size is going to allow it to be faster, propagating a nervous signal. This is really important for squid, especially because it is not myelinated. Okay, so axon diameter is going to correlate directly with surface area. You increase the diameter of the axon, you're going to increase the surface area, and therefore you're going to increase the speed of the nervous impulse. Why do you think that's the case? What happens when we increase surface area? Well, we increase membrane, and then we increase proteins. So the proteins that are going to be moving ions in and out of the axon and the sodium-potassium pump among the other protein channels, they're going to be more numerous if you have a larger diameter and you have more membrane. So the signal can go faster because we have more membrane proteins to move things in and out of. Okay. Look at this graph here. Take a minute. Notice there's 2 different y-axes here. So this one's in microns, so this one. So the blue one is for the cat axon, and the red one is for the squid. So be careful, these numbers are not applying for both. The blue for blue, red for red, okay? And then we have the velocity in millimeters per second of the conduction of the nervous signals. So take a minute and look at this diagram. So what do we see in our lovely diagram here? Well, hopefully you're noticing that there's a very different speed of these 2 neurons, right? We have the myelinated neuron for a cat, and we have unmyelinated of a squid. So which one is going faster? Well, as we increase the diameter, it has a direct impact on the increase in velocity for the myelinated cat neuron. And you'll notice here it says the cat were from the saphenous nerve and they're at 38 degrees Celsius, so this temperature also corresponds to transport, which is not what you need to know, so it should be kept constant in everything we're talking about for this course. And the unwebbed axons are from a squid at 20 to 22 degrees Celsius because the squid lives in colder temperatures, evolved for colder temperatures. So myelinated always has a higher conduction velocity. It's always going faster on the unmyelinated, even if the unmyelinated is 100 times bigger in diameter. Now look at this. This one is 800 microns at 8 microns, and it's still faster. 100 times bigger, myelin makes that big of a difference. So for some reason, the squids haven't evolved to have myelin on their nerves, on their axons, but cats and the mammals have. We don't have it on all of our nerves, just the ones that are really important. We need to be very fast, but it makes a huge difference, okay? And the IB wants you to see the difference between diameter increase and myelin increase. So try again. Take a look at this image and the two graphs and figure out what's more important for nerve impulse speed. Is it axon diameter or myelination? And use the data to help you. Okay, so you can see here just axon diameter, no myelination. Here we are, we're adding myelin in the dark gray. How is that affecting it? Look at the actual numbers and then look at the graphs as well. So we can see that if we increase the axon's diameter, it's going to increase the speed, but not proportionally. It's proportional to the square root of the axon diameter. So take the square root of 4, I get 2. So 2 meters per second is the maximum velocity I can get of an unmyelinated neuron if the axon is 4 millimeters in diameter. Whereas if I have myelin and it's 4 millimeters, now I'm going 4 meters per second. It's a directly proportional relationship here. So these kind of words are very helpful to practice. Directly proportional, proportional to the square root. We recognize that shape of that curve. But the idea is that while diameter does increase transmission speed, not nearly as much as myelin does. Okay, and HLs, you'll learn why in our next lesson. Okay, key ideas. Take a minute, write them down. If you have any questions, make a note to ask me or a classmate. All right, it's a little video. I think I'm going to show it to you guys in class so we can go through this review together, but I'll try to link it on Schoology for you as well, okay?
How?
All right, so the next part of this is synapses, so going from electrical to chemical signaling. So once we have that action potential going from negative and positively charged, it's happening inside the axon. It gets to the end of the neuron. So here is axon inside here. We have going from negative at rest to being positively charged. It gets to the end here, terminal buttons, and it's going to reach the synapse. Synapse is this part, is the presynaptic neuron, its terminal button. We have the dendrite of the next neuron here, the receiving neuron, and the little gap between is called the synaptic cleft. The synapse is all three of those parts together. So all three of these parts make the synapse. You have to have all three, okay? And notice how nothing is actually touching. It is really close, but not touching, okay? So it connects one neuron to another. It can also connect one neuron to like a motor, to a muscle cell, if it's a motor neuron, or to a gland cell, but they're going to be very, very close from the axon terminal here to the dendrite here, but it's not going to actually touch. So here's axon terminal, put purple star for it here, and the dendrite here is this side. Oops. Yeah, dendrite's here. Okay? And this is the gap between. So what's happening at the synapse is the electrical signal we have down here at the end of the axon is now going to become chemical. You can see these little brown dots. That's a neurotransmitter. It's a chemical signal. So it goes from electrical to chemical at the synapse. So here's some example of different kinds of neurotransmitters. These are the chemical signals. You've probably heard some of these before. For example, you've probably heard of serotonin or dopamine, adrenaline. The one you guys need to know for the IB is acetylcholine, because that one has to do with muscle movement. So this one is important at all neuromuscular junctions to have a connection between a neuron and a muscle, to allow a skeletal muscle to move, as you're writing notes now. But that's the only role. That's one you have to know. If you want to learn another one for another example, go for it. But the one you have to know is acetylcholine. Okay, so make a note. Okay, so this is your back to your foldable diagram here. You have this part on your diagram. We're going to go through the steps now of how a synaptic transmission goes through. Please color in, write in notes, your diagram, so make them good because you're going to be happy you have good notes for your exam in June, for unit tests coming up, and of course for IB exams and mocks next year. Remember, future you thanks current you if you take good notes, okay? So here we go. Step one, actually just going to sort this stuff in your boxes. The action potential, which is our positive charge, right? That's going to arrive at the axon terminal and depolarize the presynaptic membrane. So it's going to make the membrane positively charged as well. Normally it's polarized, which means it's negatively charged. This is going to cause voltage-gated calcium channels. So voltage-gated means that it's a protein channel that opens when a certain electrical charge passes through. In this case, it's the plus 40 millivolts charge, causes calcium channels to open, and then calcium enters the neuron. Okay. Again, if you look at your picture, you can see all this stuff. And the presence of calcium within the neuron is going to activate these vesicles of a synapse. And where vesicles are just loops of membrane that carry things inside. In this case, these vesicles are carrying neurotransmitters. So here, go back to our picture here for a second. We just talked about is here, the estradiol arrives, it's a positively charged, plus 40 millivolts, that cause these calcium channels to open, which are voltage gated. They open at plus 40, and calcium goes in, and that causes these vesicles that are carrying neurotransmitters to wake up and start moving. Okay, so again, finish your notes. And next steps. So the vesicles which contain a neurotransmitter, in our case, the only one you have to memorize is acetylcholine, and it's going to migrate to the membrane and fuse with it. And this allows it to release neurotransmitter into the synaptic cleft. Okay. Step 6, vesicles are going to release in the cleft through exocytosis. Remember, exocytosis is where the membrane of a vesicle fuses with the cell, and that causes it to release things. So release the vesicle this way. And then through these neurotransmitters, again, in our case, we're talking about acetylcholine is going to go across the cleft, that small gap of synapse, binds receptors that are specific to it. So in this case, it has to find an acetylcholine receptor, and then it's going to either excite or inhibit the membrane. Acetylcholine is going to excite it. Its goal is to make a muscle contraction happen. So if we look here, we just looked at is we have here, it's going to fuse. Here's the vesicle has fused with the membrane, releasing neurotransmitter. In this case, acetylcholine is our example. It's going to bind these receptors on the neuron's dendrite, the postsynaptic neuron, and they bind that to fit properly, and that's going to cause a response in this cell. In this case of acetylcholine, it causes it to excite, and you're going to do a muscle contraction. And that's our signal. It's already happened. The only thing we'll talk about is how we end the signal, because having a continuous signal is just as bad as having no signal at all. You need to send the signal and then stop it. Okay? So, not all neurons have all these things, but some we have that are called autoreceptors. And these are going to bind, neurotransmitters will bind to them as well when they're released in a bit of a feedback loop that's going to make sure the neurotransmitter that comes out of the presynaptic neuron is enough to send the signal but not extra. Okay? So the more the autoreceptors are bound, they're full of neurotransmitter, the less it'll release more neurotransmitter, which is good because you want the signal to be complete but not get too long. So if there's extra neurotransmitter, it takes longer to kind of clean up and end the signal, which is not ideal. And the other thing with that is we want to not waste our neurotransmitters, right? Next thing you'll notice here that happens is we have transporter proteins, and these are going to help with what are called reuptake. So we want to recycle that neurotransmitter, not make it again every single time. So we take it back up into the presynaptic neuron again, and also helps to reverse the effects. Remember, we want to have a quick, complete, clear signal and then end it. So we're ready to reset for the next signal. And the last one, enzymes. The enzyme for acetylcholine is called acetylcholinesterase. Yeah, I know it's a mouthful, guys, sorry. And it's going to be in the synaptic cleft, and it's going to break down the neurotransmitter into acetate and choline, for this particular example, as a way to stop the signal even more rapidly. For muscle contraction, which is what acetylcholine controls, makes a lot of sense because you want to have a quick movement and then stop. You don't want to have any like dragging on or laid out. So we want to have it going very effectively and signaling and then stopping, signaling and then so the next one can go. So it makes sense to try to clean it up a little bit quicker that way. So here's our diagram again. Hopefully now you have it all filled out. We have here acetylcholine coming out, It's going to bind. Here's the autoreceptor. It's going to binds. If these are all full of neurotransmitter, the cell will stop releasing extra and just like give the signal is good, there's enough signal, I'm not going to waste any more doing that. If these are empty, it'll send more. Then we have reuptake transporters. They help membrane proteins that help bring the neurotransmitter back into the cell to get reused to the next signal. And we have enzymes. If this was acetylcholine, it'd be acetylcholine esterase that breaks it down to help be able to recycle and stop even more rapidly. Because as soon as that neurotransmitter binds receptor here, it's going to send a signal. We want to send that signal, but then stop. Okay, I hope that helped. Last little bit here is we have the 3 different kinds of synapses. So we have, this is the most common one, between one neuron and the next. See how we have these synapses are connecting on the other neurons, dendrites and cell body. This is a big part of how our brain works. We have neurons to muscle, so neuromuscular junction happening here, which HLs have learned about with their muscle movement topic, and neuron to gland cells. So it could be like, for example, the gland of the pancreas releasing insulin. Right, your hypothalamus tells it to do that, and this is how it does that. Just to get some context here. Okay, last key ideas, and that is your lesson. So hopefully you guys need to slow me down. You can always do so with YouTube. You can change the talk speed, add subtitles, everything to do. I hope this is helpful. Sorry I couldn't be there today. Not feeling very well, but I managed to put this together. So hopefully that's helpful. And yeah, take care, and I'll see you next class. HLs who didn't finish your reading, this is for you next class. Building on this, okay? Talk to you later.